In this section of our notes, we're going to be talking about DNA replication. Now, to give you an idea of what you need to learn from this lecture, I've put your learning targets on Classroom, and I'm highlighting them as they've been covered. So the ones that you see there in pink, outlined in pink, are the ones that you covered with Miss Dace. And then today, we're going to be talking about these ones down here that are more of this orangish color. When you're done with this lecture, you should be able to answer or be able to talk about learning targets 12 through 16. Now you know a little bit about DNA replication because we talked about it in the last unit with mitosis and meiosis. We learned that this stage of the cell cycle, that S phase, that S stands for synthesis, and what is being synthesized is DNA. So what is happening is the cell is copying its DNA or it's replicating it. So DNA replication happens to the DNA in the nucleus during S phase of the cell cycle. And the reason why the cell is copying its DNA is because when it gets to here, it's going to split in two. So you need a set of DNA chromosomes to go into one daughter cell and a set of DNAs to go into the other daughter cell. So we are copying our DNA prior to cell division. And that's the only reason why we would ever copy the DNAs, because the cell has gotten a signal to divide, so replication is going to begin. There's a lot of enzymes that are involved in copying DNA, but we're just going to talk about two of them in pre-AP Bio, but there's going to be more that you're going to learn about whenever you come to AP Bio, but you'll have a good foundation of DNA replication. The first enzyme you need to know is called helicase. If you remember, enzymes are proteins, and we name them, we put ACE on the end of them to represent that they're enzymes. So we have helicase, and DNA is shaped as a double helix. So we're going to see helicase is going to unzip the DNA. It's going to unzip the helix. And it's going to unzip the DNA by breaking apart those H bonds that attach those two base pairs together. So it's going to break the H bonds between A and T and G and C along that DNA molecule. The second enzyme you need to know is called DNA polymerase. In the last unit, you learned that DNA is made of monomers called nucleotides that are strung together into a big polymer of DNA. So DNA polymerase is the enzyme that's actually going to put these nucleotides in order so that we can make a DNA molecule or a DNA polymer. Now I want to take a second to show you a very simple video of what these two enzymes are doing. And I'm hoping that it's going to play okay as I record it here on the smart board. Here we have a DNA molecule, so we have a DNA double helix, and we need to replicate it. The cell got a signal to divide. So the cell is going to enter S phase, and it's going to copy or replicate its DNA. We're going to go from one double helix to two double helix. Here is our molecule, our protein, our enzyme, called helicase. You can see that it's breaking apart the base pairs. So it's breaking those hydrogen bonds that are holding those base pairs together. And you're going to see two DNA polymerase molecules. The DNA polymerases, and I'm going to pause here to talk about what you see here. So these little things that are flying in here, those are free nucleotides that are found in the nucleus. And nucleotides just can't come together. So for example, if there is a T here, then we cannot just have an A nucleotide or a nucleotide with adenine just fly in and hook up to that nucleotide that has thymine on it. The enzymes have to position the two bases together so they form a hydrogen bond. So our enzymes, DNA polymerase, are going to move along the DNA molecule, and they're going to put those nucleotides together. So we just went from one ladder to two ladders. We replicated DNA. Now, one thing that you notice is those DNA polymerases, they moved in opposite directions, and that's going to be important. We're going to talk more about that here in just a minute. Moving back to our notes here. One important fact that you're going to have memorized is that when we go to add nucleotides after we split apart the DNA, we can only make a strand of nucleotides in the 5' to 3' direction. And because we can only make new strands in a 5' to 3' direction, then we're going to make these strands, we're going to build them moving in opposite directions. I'm going to start with this diagram here. You might write small. It's going to get a little confusing here with all these labels that we're going to put on this diagram. Now let's just start with our original DNA molecule, and you can see it here. So the purple part is the parent strand of DNA. Sometimes it's called the parent strand. You can call it the original molecule of DNA. This is what we're going to copy. And we can see that the DNA has already been split. As a matter of fact, let's go ahead and draw on helicase here. So I'm going to kind of put a blob here to represent this enzyme called helicase. I'm writing small for a reason. Before you try to figure out anything about replication and the direction that we're going to move these enzymes, you first need to figure out the labels, the 5' and 3' ends of our DNA molecule. So this is saying that this end is the 5' end. So if you follow this down, this purple strand down, that means that down here, this is the 3' end of a nucleotide. So that means that this end is going to be 3', and if you follow this down, this end is going to be 5'. What I tell students to do is after you split apart the DNA molecule, then I would make kind of a line, some dashed lines here to represent where we're going to put new nucleotides so that we end up with double-stranded DNA here and double-stranded DNA here. So add in those dotted lines. Now for those dotted lines, let's figure out what end would be the 3' end of the dotted line and what would be a 5' end. Well, if this end of the DNA is the 3' end, then that means when we build our new strand, the new strand will have a 5' end right here, and its 3' end, which is already labeled for you, would be down here. Well, let's take a look at this bottom strand that we dotted. If this is the 5' end of the parent strand, when we build a new strand of nucleotides, this end is going to be the 3' end. So that means this is going to be the 5' end. Let's add some arrows so you understand the direction in which we're going to make these new strands of nucleotides. I mentioned up here you need to have memorized that when we make a new strand of DNA, it's always going to be in a 5' to 3' direction. So that means that DNA polymerase is going to hook on here, and it would move this away, adding C's and G's and T's and G's and A's as it matches up those bases to the bases on the parent strand. So DNA polymerase, let's go ahead and label that. DNA polymerase, I'm going to abbreviate, DNA polymerase attached here, and it's moving in this direction. Now, some other terms that you need to know that I'm going to talk about. This area right here where it opens up, I'm just going to kind of redraw it down here. This area is called the fork, or sometimes it's called the replication fork. And you can see it labeled for you on this diagram right there. So the replication fork. So we say that DNA polymerase, when it's replicating DNA, is moving towards the replication fork. Now, let's go back to helicase here. You can probably figure out that helicase would be moving in this direction, breaking apart the hydrogen bonds between these bases. And we also just said that DNA polymerase is going to attach here, and it would add these DNA nucleotides that you see here moving in this direction. It's going to follow helicase. So think of these as like two vehicles following each other the same direction. Helicase is going this way, and DNA polymerase is moving this way as well. Because we're just going to continue to follow helicase as it unzips even more of this DNA. This strand that we're making here is going to be made continuously. We don't have to stop. We're just going to follow helicase. This strand has a name. It is called the leading strand. So let's fill in some information here. Since we have to make this strand of nucleotides in a 5' to 3' direction, and since we're going to follow helicase, we can continuously, that means we don't have to stop, we're going to continuously make this new strand of DNA following helicase. We're going to move DNA polymerase towards the fork. This is called the leading strand. The bottom strand in this situation has a different name. We're going to call it, this is the lagging strand. Remember I said when we go to build a strand of DNA nucleotides, we have to move in a 5' to 3' direction. So what's going to happen is DNA polymerase, and you see it right here. So let's label DNA polymerase. DNA polymerase is going to attach here. You can see it coming in this direction close to the fork. So it's going to attach close to the fork, and it's going to build this new strand 5' to 3'. So draw an arrow because we know it's going to be made 5' to 3'. DNA polymerase is going to attach here, and it's going to move away from the replication fork. And we're going to get just a little fragment of DNA replicated. So this part of the lagging strand, this little piece that we're going to make, because we're going to make another piece here in a second, this is called an Okazaki fragment. Okazaki fragment. So you must remember, the lagging strand is going to be made away from the fork. It's going to be made in pieces or fragments, and these fragments are called Okazaki fragments. But no matter which strand, the leading or lagging, when we build a new sequence of DNA nucleotides, we always have to do it in a 5' to 3' direction. 5' to 3' direction. So let's add that our lagging strand is made in segments called Okazaki fragments. Now let's do kind of the same thing, but let's use this diagram right here. And I'm going to add some 5' and 3' labels. If I said that the DNA, the parent strand, this end was the 3' and this was the 5', you should be able to follow this down and tell me that this end of the parent strand would be 5', down here would be 3'. And we can see that we're already starting to build some DNA here. So it looks like there's some DNA. So we're going to build some DNA here. We're going to build DNA here as we match up nucleotides to the parent strand. Okay, now we need to figure out our direction. When we go to assemble these nucleotides, we have to assemble them in a 5' to 3' direction. This end will be a 3' end. This end will be a 5' end. We have to build this strand in this direction, away from the fork. So that means that we are going to hook on, so let's add, DNA polymerase is going to attach close to the fork, and it's going to move this way, away from the fork, until it gets here. Because we've already made this segment of DNA, from here to here. This is one of those Okazaki fragments. But as Helicase unzipped even more of the DNA, now we have this area right here that we need to add bases to. So we're going to make another Okazaki fragment here. So DNA polymerase is going to move until it reaches that fragment that previously was replicated. You could tell that this is the leading strand down here because there's already some arrows for you. Leading strand. Notice we have DNA polymerase here, the blue dot. We have an arrow saying it's moving that direction. It's moving following Helicase. It attached here, and it went this direction, and it's going to keep adding DNA nucleotides to the parent strand. So we have double-stranded DNA. Now, to help you visualize both the leading and the lagging strand, I have another video that I'm going to attempt to show you and record. There's a lot more to this video than what you really need to know, but I'm just going to highlight a few things here. We're going to see Helicase is going to unzip the DNA molecule. So that must mean that this cell is in S phase. So here comes Helicase, okay? And you're going to see that we have labels here. It's saying this is 5', here's 3'. So when we have a new strand built here in the middle, this will be 5', 3'. So we know this new strand is going to be made in this direction towards the fork. I hope you can see the writing there. So that means that DNA polymerase is attaching here, and it's moving towards the left here. And that's what you're going to see here in a second. So here comes DNA polymerase, and notice it is following Helicase. So that top strand is going to be called the leading strand. It's being made continuously. Okay? I'm going to back this up a little bit. Okay? So now that we have, for this blue strand, we have this much unzipped. We're going to make new DNA here. We're going to make it, here's the 3' end, here's the 5' end, in the 5' to 3' direction. So we're going to hook DNA polymerase here and build the new strand away from the fork. We're going to get fragments, we're going to get pieces. Now let me go ahead and kind of move this down so that you can see we're making that top strand continuously. That would be the leading strand. Okay? There's a lot of things you're going to see here, but I really want you to pay attention to the direction of the way we synthesize or replicate this bottom blue strand. Notice the direction that it goes, away from the fork. Okay? When we go to make the DNA double helix, notice that DNA polymerase moves away from the fork. So we just made a little piece, or right there. We just made one little piece of DNA that you see right here. That would be a fragment. Now we're going to do this part. So here comes our replicating enzymes, and so in the next little fragment, we're going to make it there, and we're going to make it away from the fork. I know there's a lot of other enzymes that it's showing you here, and you don't need to know that. But I think this is a good visual that shows you that one strand is going to be made continuously, where the other one's going to be made in pieces away from the replication fork. Now to finish up this page of notes, this model of how DNA gets replicated has a name. It's called semi-conservative replication. If you think about what we mean by semi-sweet chocolate, that means that it's chocolate that's kind of sweet. It's not as sweet as milk chocolate. It's kind of sweet. So semi-conservative means we kind of conserve the original DNA molecule. If we go back up to our diagram up here, is we kind of keep this original parent strand intact. We put half of it over here and half of it over here into these two DNA molecules. And that's what we're trying to show you here with this color code. This is the original strand, the parent strand. When we're done replicating it, half of it is going to be in this new DNA molecule, and the other half of this parent strand is going to be in this other DNA molecule. So we kind of conserve the original. We don't make an exact, we don't have these two strands read together over here as a result of replication. We split apart the red DNA strands and we build new ones off of the original strand. So we kind of conserve the original. So it's called semi-conservative replication, because one strand of each new DNA molecule is from the parent DNA molecule, or the original DNA molecule.